Ever wondered why traits follow predictable patterns from parents to offspring, and how that ties directly to what's happening in meiosis? The chromosomal theory of inheritance is the bridge between Mendel's ratios and the physical behavior of chromosomes. In this video, we'll connect genes to chromosomes, track how they segregate and assort during meiosis, and show how this explains classic inheritance patterns you'll see on the MCAT. By the end, you'll be able to predict outcomes with confidence and avoid the most common genetics traps. The chromosomal theory of inheritance states that genes are located on chromosomes, and their behavior during meiosis explains Mendelian inheritance patterns. This idea, developed in the early 1900s by Sutton and Boveri, bridges the gap between Mendel's abstract factors and physical structures we can observe under a microscope. Chromosomes carry genes, and because chromosomes segregate and assort during meiosis, genes follow predictable inheritance patterns. In meiosis 1, homologous chromosomes separate, which directly corresponds to Mendel's law of segregation, where allele pairs split so each gamete carries only one allele. Then, in metaphase I, homologous pairs line up randomly, explaining independent assortment of genes located on different chromosomes. A helpful mnemonic is Sutton saw segregation, linking the scientist to the concept. On the MCAT, you may see questions asking you to connect stages of meiosis with inheritance patterns, or predict offspring genotypes based on chromosomal behavior. This concept also ties into non-disjunction, where improper chromosome separation leads to disorders like Down syndrome, reinforcing that chromosome behavior directly impacts inheritance. Understanding that genes are physically located on chromosomes turns Mendelian genetics from abstract ratios into a concrete, mechanistic process. Homologous chromosomes carry the same genes at the same loci but may have different alleles, which explains variation in inherited traits. Each homologous pair consists of one chromosome from each parent, meaning you inherit two copies of every gene, one maternal and one paternal. These genes occupy identical positions, called loci, but can differ in their sequence, giving rise to different alleles such as dominant or recessive forms. This structural organization is key to understanding why individuals can be heterozygous or homozygous for a trait. During meiosis, homologous chromosomes pair up and can exchange segments in a process called crossing over, which increases genetic diversity. A useful way to remember this is same genes, maybe different versions. On the MCAT, you may be asked to determine how allele combinations lead to phenotypes or how recombination affects genetic variation. This concept also connects to linkage and recombination frequency, since genes on the same chromosome tend to be inherited together unless crossing over separates them. Recognizing that homologous chromosomes are similar but not identical helps explain why siblings can inherit different combinations of traits despite having the same parents. The law of segregation is explained by the separation of homologous chromosomes during meiosis 1, ensuring each gamete receives only one allele per gene. During meiosis 1, homologous chromosomes are pulled to opposite poles of the cell, which physically separates the two alleles for each gene. As a result, each gamete ends up with only one version of each gene, restoring haploidy. When fertilization occurs, the diploid state is restored, and allele pairs are re-established. This mechanism directly supports Mendel's observation that traits segregate in predictable ratios, such as the classic 3:1 dominant to recessive phenotype ratio in monohybrid crosses. A quick mnemonic is segregation equals separation. On the MCAT, you might be given a meiosis diagram and asked to identify when alleles separate or predict outcomes if segregation fails, such as in non-disjunction events. This concept also connects to gamete formation and Punnett squares, which rely on the assumption that each gamete carries only one allele. Understanding the physical basis of segregation helps you move beyond memorizing ratios to actually predicting how genetic information is distributed. The law of independent assortment arises from the random orientation of homologous chromosome pairs during metaphase 1 of meiosis. When homologous chromosomes line up at the metaphase plate, each pair aligns independently of the others, meaning the maternal or paternal chromosome can face either pole. This random alignment leads to a variety of possible chromosome combinations in gametes, greatly increasing genetic diversity. For example, with just 2 chromosome pairs, you can get 4 combinations, and in humans with 23 pairs, the number of combinations is over 8 million, even before considering crossing over. A helpful mnemonic is independent equals random arrangement. On the MCAT, questions often test whether you understand that this law applies only to genes on different chromosomes or those far apart on the same chromosome. Genes that are close together are linked and do not assort independently, which is a key exception. This concept connects directly to probability in genetics, where you multiply probabilities of independent events to predict genotype frequencies. Understanding the chromosomal basis of independent assortment allows you to reason through complex inheritance patterns rather than memorizing outcomes. Linked genes are located close together on the same chromosome and tend to be inherited together unless separated by crossing over. Unlike genes on different chromosomes, which assort independently, linked genes travel together during meiosis because they are physically attached on the same DNA molecule. However, during prophase 1, homologous chromosomes can exchange segments in a process called crossing over, which can separate linked genes and create new allele combinations. The closer 2 genes are, the less likely a crossover will occur between them, which is why recombination frequency can be used to estimate genetic distance. A useful phrase is linked means locked, unless crossed. On the MCAT, you may encounter problems involving recombination frequencies or genetic mapping, where you calculate distances in centimorgans. This concept also connects to pedigree analysis and understanding why some traits do not follow classic Mendelian ratios. Recognizing the difference between independent assortment and linkage is crucial for interpreting more complex inheritance patterns and for understanding how genetic diversity arises at the chromosomal level.